everybody, and you're currently watching Daily News with Ruhaan. And surprisingly enough, today we don't only have daily news on our show. However, we have prepared an exclusive segment, an exclusive interview arranged with someone you'll know later about a groundbreaking news which is currently only exclusive to our show. So stay tuned and watch these ads and don't leave. Welcome back, everyone, and this is the exciting news we're talking about. A groundbreaking research has been made on genetic engineering and gene editing, and today we have invited the lead scientist of that research, Mr. Bhargav, to our show to explain to you guys how it works and how it could impact your and your kids' futures. So here is the segment coming up with Dr. Ruhaan. Hello everyone. This is Professor Bhargav reporting from the lab which has made the great breakthrough in genetic engineering and gene modification over the past few years, I must say. So yes, today the topic of our discussion would be gene editing, and to understand what breakthrough we've made and how it could impact your lives, we really need to understand what gene editing is. The basic definition, to wrap your heads around what gene editing is it's the artificial modification and recombination of DNA or other nucleic acids in order to modify an organism or living being and their populations. So there might have been some words in there which might have confused you. For example, what's DNA? How do you recombine and how do you manipulate it, and what's nucleic acids? So let's start by understanding the concept of nucleic acids. Nucleic acids are basically a combination of nucleotides. Nucleotides are made out of three main products. There are 5-carbon sugar, a phosphate group, and nitrogen base. And nucleic acids basically are a generalized name for DNA and RNA, and there's only one change between nucleic acids which classify them to be either DNA or RNA. DNA, deoxyribonucleic acid, is in which the carbon sugar is derived from the ribose. However, RNA is when carbon sugar is compound ribose. So this basically differentiates how they're made. But what basically are DNA and RNA? Well, DNA is a nucleic acid containing genetic instructions used in development and functioning of all known living organisms. The DNA segments carry this genetic information are called genes. And what's RNA? RNA, ribonucleic acid, functions in converting genetic information. So basically, it functions as a text converter. It can change your Word file into PDF. Similarly, it changes the genetic information from DNA into messenger RNA, which can be read by proteins across the body. So now, we're not necessarily interested in RNA as much as we're in DNA, as the process of genetic engineering is to manipulate your genes, which are constructed out of your DNA. So to understand how big of an achievement we have made in this genetic engineering area is to understand what basically is genetic engineering, and what better way to do that by looking at the history of it. So basically, this first idea of gene editing emerged in Switzerland by this person called Werner Arber, who developed this technology, this chemical which would eliminate random strands of information from the DNA. However, this was not very good as it eliminated random strands. This idea was then further purified in America the latter year by Hamilton O. Smith, which created a technology which could basically pinpoint a certain gene and eliminate it. However, it didn't necessarily function like that, as there was a very low success rate, and that chemical could also go wild and eliminate random genes. And so this basically developed over the years, and now the genetic engineering as we know it in the 21st century is basically CRISPR Cas9. And CRISPR Cas9 is very interesting as it as it takes previous ideas, gives it a new definition by using parts of our own body to modify itself and transform what we know as randomness into basically structural and developable genes and information that basically makes us. And you might be wondering what CRISPR-Cas9 is, as this is basically gene editing 2000s. CRISPR-Cas9, you might think, is a system. However, it stands for clustered regularly interspaced short palindromic repeats, and it's not necessarily a system. However, it's a process our body follows. So, on the principles of natural selection, there are some species that survive nature and that survive certain predators and natures. And while surviving these, our body develops information that can be used to retaliate against these particular predators or natural instances if they were to happen again. This stores in our DNA, and that portion of the DNA is known as CRISPR. And Cas9 is basically a protein which, when any turbulence is detected in our body, takes that CRISPR from our DNA and uses it and matches it with every single piece of information, every single DNA in our body to see where the defect is so it could eliminate that. And what CRISPR-Cas9 does is basically uses Cas9 and manipulates it by injecting RNA, mainly messenger RNA, which carries all the information of the RNA, into that Cas9 so that it could examine our whole body for that unwanted gene and eliminate it. That was my basic definition of what CRISPR-Cas9 is. Now I shall explain to you what we have achieved in this year. So basically, we have advanced our understanding of CRISPR-Cas9, and we have made solutions and formulae of diseases such as HIV and cancer. And this big achievement, as now we can inject these RNA and these DNA sequences of corrupted cells like HIV and cancer into Cas9, which would then further look into our whole body for such cells which are infected or taken over by cancer and HIV and eliminate them. And these are, mind you, diseases that humans have always suffered with, and some people and some societies have given these diseases names such as the greatest opposition of mankind. And we have actually confirmed a successful test of HIV cells in rats. So we took a rat whose approximately 99% of its cells were infected with HIV. However, after injecting and processing this HIV cell into Cas9 and using the CRISPR method, half of such cells were then reused, half of the cells eliminated HIV viruses, and the fascinating part is that none of those cells died, hence this ensured that the species will survive. So now we can use this and further implement this technology into humans, and maybe, just maybe, there's a high likelihood that using this technology we can eliminate major diseases and untreatable diseases like cancer, HIV, and viruses such as Ebola from the face of the planet. Thank you for this explanation, Mr. Bhargav, and I must say, oh my god, I'm extremely fascinated and excited about this new technology, as this could eliminate one of the highest population decreasers in our planet and one of the most frequent and the most painful killers of mankind, diseases such as cancer and HIV. However, everyone, Now that Mr. Bhargav is gone, we need to talk about the other side of this concept of genetic engineering because we want to report to you all different sides of the argument. Now, after this news was released into media and to us, there have been certain oppositions by certain scientists across the globe. One of them, a very prominent one, which we have invited today to discuss why he thinks and what he thinks about the ethics behind this new discovery of gene editing and gene manipulation and how it can be implemented to our society with such cheap costs and in such recent times. Uh, so stay tuned and wait for the interview. Hello everyone, we're back and we have the professor, the scientist online with us. And he is today going to explain to us what he thinks basically are the ethics behind this concept of genetic engineering and why this is not necessarily all good and what this can actually transform into as time comes and as it becomes more affordable for everyone to get these treatments. Are you live? Okay, thanks. Thank you, Mr. Ruhaan, for inviting me to your show and allowing me to discuss basically what I feel about this breakthrough in genetic engineering and how everything, especially in human hands, which comes out as good, might not always result in the way we expect it to. Now, don't get me wrong. This concept of genetic engineering and gene editing will bring end to like massive diseases such as cancer and HIV, which have been untreatable and defined as undefeatable in our society since like the dawn of human existence and have been one of the major killers of mankind. However, as this technology develops, the main goal of businesses is to always make it affordable for the people, right? And gene editing is a very powerful tool which can be used to do other things except treat diseases and eliminate viruses in our bodies. It allows for parents to have a say in how their offspring would function, how he or she would look like, what gender he or she will be, what eye color, what hair color, mental strength, cardiovascular strength, organ strength, muscle strength, basically everything. And such a tool, mind me, will, as much as it will bring joy, technically, and bit more of a control over humankind in our society, will form a uniform society, and there will be no room left for individuality and imperfections, which humans have actually taken advantage of over since everyone existed, as not everyone was good at everything, and there were people who were better at certain things than anyone, and you as a person always had something that you were better at doing than anyone you had ever met. You had these unique characteristics combined into you to form a person that you are or a person that I am. However, genetic engineering will allow humans to take that away from mankind, and then every kid will be the fastest kid, every kid will be the smartest kid, and everyone will be basically good at everything. So that will not only take away from the concept of individuality, however, it will increase competition in all parts of society, which will make everything just more expensive and economically depriving, as governments will have to pay their citizens more because everyone's just putting so much effort into everything. And then the taxes will have to be high, and then this might even lead to unhappy societies, who knows. However, even though there are so many negative aspects to it, we might also question the basic ethics of it. Should you allow the everyday man to actually have a say in who is coming into this earth, how he or she will look like, instead of nature deciding that instead of natural processing taking care of that? And will you, as a person, I'm talking to everyone in this concept, will you, as a person, feel comfortable with being a product of basically genetic manipulation and um, so talking to everyone here, will you like to have your genes taken away because your parents fancied something else in you rather than who you actually are? Will you find it right for you to have your kid to have what you want him to have rather than him to be an individual and an interesting personality? I mean, there are many arguments for and against this concept of genetic engineering. However, I feel like it's a great advancement. However, such a thing needs to be kept in control and kept in the hands of professionals, as I say, instead of having let it out in public just for profit reasons. That it could cause massive damages to human society as we know it, and just lead to increase in competition, pressure, and basically just an increase in this idea of utopia which can never be achieved. Thank you very much. This was all I had to say about this topic. So thank you everyone for watching this video, and this was basically all we had for daily news with Ruhaan today. Don't forget to leave your opinions about this debate on genetic engineering and whether it should be pursued further or not on our socials, on our blog, on our Twitter. Thank you very much for tuning in, and this was all we had for today. See you.